Hi, we're continuing with life science and cell biology in a nutshell. So currently we're learning about a molecule called a glycolipid, which consists of a glycerol backbone, um, fatty acid tails, just like in phospholipids. However, it has sugar molecules bound to the glycerol backbone, making it a glycolipid. Um, if the sugar that is bound to the backbone is uncharged, then the molecule is uncharged. If it's negatively charged, this is called a ganglioside molecule. So it just depends. There may be different kinds of sugar molecules, such as galactose, glucose, etc. Sialic acid, different kinds of sugar molecules that may be bound to the backbone, or acetylgalactosamine, which is GalNAc short. So their localization in the cell membrane helps protect the membrane against harsh conditions such as low pH and degradative enzymes. Um, Charged glycolipids also may be important for their electrical effects, altering the field of across the membrane and the concentrations of ions such as calcium at the membrane surface. They also function in cell recognition processes, um, in which membrane-bound carbohydrate-binding proteins called lectins bind to the sugar groups on the both glycolipids and glycoproteins in the process of cell-cell adhesion. Surprisingly, however, mutant mice that are deficient in all of their complex process um, of complex gangliosides show no obvious abnormalities, although males cannot transport testosterone normally and are consequently sterile. Whatever their normal function, some glycolipids provide entry points for certain bacterial toxins. The ganglioside GM1, for example, acts as a self-surface receptor for the bacterial toxin that causes the debilitating diarrhea of cholera. Cholera toxin binds to and enters only those cells that have the ganglioside GM11 on their surface, including intestinal epithelial cells. Its entry into a cell leads to a prolonged increase in the concentration of intracellular cyclic AMP, which in turn causes a large efflux of sodium and water into the intestine. So Na plus and water into the intestine. So biological membranes consist of a continuous double layer of lipid molecules in which membrane proteins are embedded. This lipid bilayer is fluid, with individual lipid molecules able to diffuse rapidly within their own monolayer. The membrane lipid molecules are amphipathic, which means they are polar and nonpolar. The most numerous are the phospholipids. When placed in water, they assemble spontaneously into bilayers, which form sealed compartments that reseal if torn. There are three major classes of membrane lipid molecules: phospholipids, cholesterol, and glycolipids. So, cholesterol is a steroid molecule and serves to add rigidity to the membrane. Glycolipids help with cell signaling. Um, so they are a lipid and a sugar molecule. Um, together. The lipid compositions of the inner and outer monolayers are different, reflecting the different functions of the two faces of a cell membrane. Different mixtures of lipids are found in the membranes of cells of different types, as well as in the various membranes of a single eukaryotic cell. Some membrane-bound enzymes require specific lipid head groups in order to function. The head groups of some lipids form docking sites for specific cytosolic proteins. Some extracellular signals that act through membrane receptor proteins activate phospholipases that cleave selected phospholipid molecules in the plasma membrane, thereby generating fragments that act as intracellular signaling molecules. Here you see the extracellular space, the lumen of the cell, the inside. You see a diagram of the phospholipid, which comprises the majority of the lipid bilayer. Well, it's a two phospholipid molecules facing one another comprise one monolayer. And in eukaryotic cells, cells are surrounded by a single bilayer. So two rows of facing phospholipid molecules. In bacterial cells, you have two bilayers, so four rows of phospholipids facing each other. And this is because the second layer in bacteria acts like a cell wall for extra protection. In eukaryotes, however, it's just one bilayer, and the phospholipids have with them cholesterol and glycol lipids, as well as various proteins, which serve as channels and receptors as well inside the membrane. So the various molecules function for cell interaction, signaling, etc. Various functions. Phospholipids are their class of lipids, which is the major component of all cell membranes. They form the lipid bilayer because of their amphipathic characteristics. So they're nonpolar tails, fatty acid tails, or hydrophobic tails, which face one another because the nonpolar tails interact, and the polar hydrophilic heads face the water and the solution, which is polar, and they interact with other polar parts of other molecules. So two hydrophobic fatty acid tails and a so there's two tails, one being straight, saturated with no kinks, all single carbon hydrogen bonds, and the second tail is an unsaturated chain which has a kink, meaning there's one double bond between two of the carbon hydrogen groups, creating the kink. So the fatty acid tails, two of them, they're not lined up perfectly, they're not perfectly symmetrical, which contributes to the fluidity of the membrane because it reduces the stacking of these phospholipid molecules. So they're not as rigidly close together. And so the head group of the phospholipid and the tail group are joined together by a glycerol molecule or backbone. The phosphate group, which is in the polar side, the head group, can be modified with simple organic molecules such as choline and various alcohol groups. As we learned earlier, there's four different kinds of phospholipids. Two are usually found in the outer layer and two are found in the inner layer.